Okay, good afternoon, everyone. For those who don't know me, I'm Ece. I'm one of the affiliate faculty at CCNB. We are hosting Dr. James Zhou today. Dr. Zhou is currently an associate professor of biomedical informatics and computer science at Stanford. He received his PhD from Harvard in 2014. And after that, he was a member of Microsoft Research and a Gates Scholar at Cambridge and a Simons Fellow at UC Berkeley. He joined as a faculty at Stanford in 2016, and he's a Chan Zuckerberg investigator. He has received 2 Chan Zuckerberg Biohawk Investigator Awards, Sloan Fellowship, and Secretary Award, a top 10 clinical achievement award, and faculty awards from Google, Adobe, and Amazon. His research focuses on making machine learning more reliable, human competitive, and statistically diverse. Please join in welcoming Dr. Zhou. Thank you.
Okay, great. Well, thank you very much. It's wonderful to come to visit Brown, and really nice to meet many folks here, and a lot of fun conversations during the day. So yeah, so I'm excited to explore some of the recent directions that we're doing around building AI agents to really help to automate and to accelerate different kinds of biomedical and scientific discoveries. So the context of this is that, as you know, so AI has been very useful as a tool in various scientific and biomedical research, right? So usually the way that we use AI is that if there's a well-defined problem, right, then we can design a specific AI algorithm to tackle that particular problem, right? So for example, if there's a problem of trying to predict structures of proteins, then you can use tools like AlphaFold, which would be very powerful for solving that problem. So there's sort of a paradigm shift over the last couple of years, where instead of thinking of AI as a tool for solving a well-defined problem, now people are starting to use AI as a sort of a partner or a co-pilot to try to tackle a broader range of research endeavors. So this ranges from using AI to help us with generating hypotheses, writing papers, writing code, or even try to do the experiments themselves. Right, so a lot of that advances are driven by the advances in what people call AI agents, which are basically systems that you build on top of large language models like ChatGPT, whereby you endow the systems, these models, right, with the ability to use different tools like AlphaFold. But then these large language models can also communicate with each other and also communicate with humans, right? So in that sense, then they're not just like a narrow tool for solving a specific problem, but can be potentially like a co-pilot across this broader research endeavor. So I think it's still quite early stages of these AI scientist agents, but there's a lot of exciting developments. So that's what I want to discuss today, is how we can think about building these AI scientists, what are different ways of interacting with these different AI scientists, and what are their limitations, and how that could potentially change how we do science and also communicate science. And this is meant to be very interactive, so if you have questions, just feel free to interrupt me at any point, so you don't have to wait until the end. So I first just want to give a few examples, concrete examples to illustrate what are the kinds of AI scientist agents that we could build and develop, and just also to give you a flavor of what they can do, the kinds of research they could do. So the first example is a system that we built relatively recently that we call the virtual lab. So it's basically a team of AI agents that work together. It sort of mirrors my physical lab at Stanford in the sense that the virtual lab also has the AI professor agent. And working with the AI professor agent, there's actually a bunch of different AI student agents with expertise in different areas like chemistry or computational biology. And they actually run their own group meetings, the agents do, right? And we give the agents some physical money, the budget to do real experiments. And the agents also, we built for the agents a school, like a virtual lab school, where the agents can actually go to the school and to teach themselves how to become a better expert in these particular domains. So I want to explain a bit of how this virtual lab works. So the way that we interact with these AI agents in the virtual lab is we mostly will talk to the AI professor agent. So for example, I can explain to the AI professor agent some high-level description of the project that I'm interested in. So for example, maybe I'm interested in in developing new proteins for this particular target, where I want to analyze this complex genomic data set, right, where I want to design a clinical trial, right? And then the AI professor agent would look at that particular project and then try to identify what are different areas of expertise that are needed to tackle that project. So maybe for this project of designing new proteins, maybe the professor agent would say, it's useful to have a computational biologist or a protein engineer on the team. In that case, then the PI agent's job is actually to create and to train the sub-agent that have that relevant expertise. So I'll come back to you on a couple of slides to explain more about how that training process happens for the agents. For now, you can think of each agent as under the hood being like a large language model. In this case, we can use ChatGPT-4 or GPT-5. And then we're going to fine tune these agents to endow them with more specific expertise and knowledge in these particular domains. And once they are created, then these agents can start to run their own group meetings. So similar to our group meetings, they can meet together and have a research discussion. They can also have one-on-one meetings where one of the agents can meet with the PI agent to review a piece of the data or analyze a particular code. Yeah.
I have two questions. One, why do we need multiple agents with different expertise instead of one agent with all these expertise?
Yeah.
And number two, how experimental is this approach versus are you using this actively in your research background?
Yeah, so both are good questions. I'll show some examples of the multi-agent interactions. And we did a comparison where we have multiple agents with different expertise versus like a single agent. We found that the multi-agent process actually ends up being much more creative and more reliable than try to have a single agent to solve the problem by itself. And as we'll see why that happens, basically what happens is that when you have multiple agents, they actually start to build ideas off each other. They also have debates and disagreements. And through that debating process, that actually led to more creative and more robust insights than if I just have a single agent to solve the project. Yeah, so good questions. So basically, the agents then will have these team meetings and then individual meetings, right? And all of that meetings will happen in sort of like a virtual sandbox environment. So the agents are able to use tools and execute code in this kind of virtual environment. Okay. And then I just want to make this process more concrete by just show you an example of what this agent's team meeting will look like. So this is basically the verbatim transcript from one of these research meetings from the AI scientist agents. This is actually basically for the first project we give the agents. So we told the agents to basically design binders for the recent COVID variants. All right, so at the time that we started this project last year, there are some quite recent COVID variants, called JN1 variant. And for these new variants, there were actually no known good binders. So this is kind of a very challenging open research project. So we thought, okay, so could we just give this very challenging problem to the agents and see, could they do something with this? So in the beginning, so you can see that the PI agent first just sort of explains the project to the other agents, right? So it explains that our goal here is to design binders to the latest COVID variants, to the spike protein, and also explains to the other agents why that's an important project. And then after that, the way that it works in these group meetings in the virtual lab is that the different agents can then speak up one after another to contribute their ideas, right? So in this case, the immunologist agent spoke up next and recommended that to design these binders in the form of nanobodies. Now, this is kind of already a quite surprising recommendation because most of the time, if you ask human researchers, they would say, let's design antibodies, right, to bind to the target. So antibodies are much more prevalent, and they're often used as a sort of a binding substrate as for vaccines and also for therapeutics. Nanobodies are basically smaller cousins of antibodies. They're only found in a few animals like camels. So it's much less common. So it's kind of quite an unorthodox recommendation to design nanobody binders rather than antibody binders. But this actually turns out to be quite a wise decision, as we'll see shortly. So the machine learning agent spoke up after the immunologist and says that, okay, I actually like this idea of designing nanobodies, and for the following reason, because these nanobodies are smaller, that makes my job as the ML agent easier to computationally predict their structures using deep learning methods, right? And then the computational biologist agent says that, okay, in addition to using these deep learning methods, I also recommend using these more physics-style molecular dynamics and simulations to study the structures of these proteins. So you can see how they can sort of complement, but also bounce off ideas of each other. And in all of these meetings, we also have a scientific critic agent. So it's basically like a reviewer agent whose job is to try to provide more skeptical feedback to the ideas of these other agents. In this case, the critic agent says that, well, if we're going to design nanobodies, there's not a lot of public data sets around nanobodies for COVID. So you need to be super careful about overfitting to the small amount of existing data. Right. So the actual meeting goes on for longer, right? They can speak multiple rounds. But here, basically, it shows the first part of the meeting, and you can already see from this transcript how the different agents can communicate and work together, build ideas off each other. They can also have, some, back and forth, and maybe some disagreements, but they can relatively rapidly sort of converge into like a concrete research plan. Yeah. So it's a good question of how we determine the orders. So in general, the order could be random, right? And the order with which scientists speak also very much affects the trajectory of the group discussion. So what we actually do is that we actually run multiple meetings in parallel, potentially with different orders. And this is actually one of the things that I'm very envious of the AI scientists, is that their meetings are much more efficient than my meetings, right? So one of their meetings maybe would take a few seconds or less than a minute. So in the time that we had one of our meetings, the AI scientists already had hundreds of research discussions. And because they don't get tired, so they actually run multiple meetings in parallel. So basically for every scientific decision that they need to discuss and make, they run 5 meetings in parallel. In each of these parallel meetings, they'll discuss the same question, but they could actually speak in different orders. And there's also randomness in their underlying language models. So in these parallel meetings, they actually often come up with different ideas. So then what we do is that we ask the PI agent to basically read the transcripts of all these parallel meetings to come to a consensus of what to do next, right? So this can, by running these meetings in parallel, then they can more efficiently explore a broader range of ideas and try to be more creative. Great. And in case people are curious, I'm showing here in this pie chart, basically how often the different participants would speak in the virtual lab discussions. So the blue part is basically for the human researchers, like for example, myself, my students, we only speak about 1% of the time, right? So we don't talk very much because our job is to mostly provide some high level feedback or guidance to the AI scientists. So for example, we'll tell the AI scientist, here's the description of the problem that we're interested in. That's why it's useful for humans. And we also tell the AI scientists some of the constraints that we have. One example constraint would be we tell them, here's how much money we have to do the wet lab experimental validations, right? So they can't go too crazy with their experimental designs. Otherwise, we mostly leave it up to the agents to come up with their own research ideas and to implement that, right? So we don't want to micromanage the AI scientists too much. For this particular project, the computational biologist agent does the most talking, which I think makes sense because the CompBio agent has a lot of work to do to basically to come up with the computational pipelines for modeling these nanobody structures. The machine learning agent here's job is to actually do all the coding for the other agents. So for example, another scientist agent would say, implement this algorithm for me, and then the ML agent will actually write the code. So this agent doesn't have to talk so much in English, but it also does a lot of work in basically writing all the code for the other agents. So any questions about how the interaction in the virtual lab works? Yeah. Yeah, so the questions around how does the persona for these different agents come about? So part of the persona comes from giving the agents these specific instructions, right? So for example, for the PI agent, then the instructions to the PI agent would be like, your job is to mostly organize these meetings, right? So you have to come up with the research agenda for every topic and assign tasks for those sub-agents. A second part of the persona comes from because these agents have different areas of expertise. And then to gain that expertise, then each of the agents then need to basically learn to use the tools within that domain. So for example, like the computational biology agent needs to be familiar with AlphaFold, right? And the machine learning agent needs to write the code, right, or the scripts. So some of that persona actually happens because of the domain-specific tools that each of these agents have access to. And then third part is that we can also endow the agents with more specialized knowledge by having the agents go into the agent school through this fine-tuning process I'll explain next. Yeah, so the humans can basically interact in the virtual lab anytime that we want, right? So we usually find it useful to first interact in the beginning by telling the agents their overall project direction, right? So we want the agents to work on projects that are useful for us. And then we can interject at different points. For example, if we see that something the agent is doing something that's not correct, then we can tell the agents, oh, have you thought about that? Or try to use a different tool. So we can interject at these different points, maybe as a part of each of these group meetings.
Well, you mentioned that a lot of times, like even when state agents interact, makes the content a lot different. It's actually like something else, like the PI agent. I was also curious if you've also looked into like as the PI, like when the PI agent recommends like an experiment, like to based on all these conversations, does the recommended like experiment to be with, does that change at all if you look at it multiple times?
Yeah, so the recommended experiment from the PI agent is typically pretty robust because it is trying to take a consensus of across the multiple parallel meetings. And that consensus, which forms the next set of experiments, is often more robust than the decision from each of the individual meetings. Yeah, but there's still some randomness in the PI agent as well. Yeah.
I have two related questions. So first off, like, I guess when I interact with ChatGPT, sometimes it wouldn't code the way I want to, or the code is wrong.
Yeah.
So I'm wondering, like, would it get stuck? For example, like if one agent throws the ball, basically. And also, I guess even if when things do work out, do we have a sense of, I guess, where the credit goes to? Like, is there one agent that does a really good job? Like, is there an agent that's basically like the weakest link?
Yeah. Yeah, so the question is first, what happens when one of the agents makes a mistake? And the second is how do we assign credits to the different agents? So agents do make mistakes, and we'll come to some of those mistakes. Here, they do have some sort of mechanism to fix those mistakes. For example, the PI agent would actually meet with the individual agent, right, to review their code or review their analysis. And in that review process from the one-on-one meetings, that's often how they do a lot of the debugging. And then for the credit assignment, I think there is kind of interesting. One thing that we have done is to basically do some ablations to try to say what happens if I remove a particular agent from the team, how does that affect final outcomes, right? And we do find that, for example, if you remove the critic agent, right, that actually made the process less reliable. There's more hallucinations, more mistakes, right? So that's how we quantify all the critics agents is quite useful in the debugging process, right? The immunologist agent is quite useful in providing some of these research plannings, right? So we do these ablations as a way to do figure out the contribution of each agent.
Thank you.
Great. Okay. So now that we see how the agents interact, I want to come back to and briefly explain how the school works, right? So the motivation for having a school for the agents is that if you take the off-the-shelf models like GPT-4 or GPT-5, so these models are quite good at more general, broad knowledge. But if you go really deep into a particular domain, right, often these models do not have the most up-to-date or the deepest insights in that particular domain, right? For example, they may not know about the very recent variants, right, for COVID or for a particular disease. Then to fill in that gap, that's why we built the school where the agents can go to the school, and it's a relatively autonomous school, so the agents can actually design their own learning curriculums and find their materials to help them to become a better expert in that deeper topic. So the way that a school works here is that, so first we will tell the agents, here are some of the topics that are important for them to learn about. So one topic could be like learn about the recent variants, or learn about the recent clinical trials, or learn about the latest tools in this domain. And for each of those topics, then the agents will actually design their own learning curriculum by coming up with a set of questions that are relevant for that topic. And then for each of those questions, we enable the agents to actually go out and then to search and download articles and papers and code that are relevant for that questions. For example, they can go to PubMed Central and find all the open source, open access articles, and then download those articles that are relevant. And then they would actually read each of those papers in some depth. As they read the papers, then when they allow the agents to basically update their model parameters. So basically what happens under the hood is that each of these agents is a large language model, so it has a transformer neural network under the hood. And initially, they all have the same neural network, right? They're all the GPT-4, GPT-5. But then as the agents actually read their own materials, right, the papers that they download, then we allow them to basically update the weights of the transformer to help them to ingest that new knowledge. So then they start to diverge. The parameters gets different now for the immunologist agent or for the comp bio agent, right? So then how they become different experts. And then after that, it won't be a very good school if there's not some sort of assessment mechanism. So then we have like another sort of teacher agents to generate quizzes to test the student agents. Right, student agents don't have to pass those quizzes. If they don't do well, they have to go back to school, do additional readings, additional trainings before they could graduate. Right, and then if they pass the quiz, then they can graduate and then come and work in the virtual lab. So this is a relatively efficient process, usually within a couple of days. Right, then that's the agent can go from start to finish, go finish their training and assessments, and then graduate from the lab school. Yeah. 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 So good question. So basically how the fine-tuning works is that, let's say the agent downloads a set of papers and they read those papers. And then from the papers, they can basically turn those papers into let's say question answers, right? Or they can also turn the papers into some summaries. And then they basically can do next token prediction to update their model parameters based on those question answers and also based on the summaries from each of these papers. Right. So essentially for that fine-tuning or SFT parts, then the models are basically updating their model parameters so that they could most accurately answer the, be able to reproduce the summaries from that paper.
AI does creates the question itself, right? So
Yeah.
if you have a like long term, for example, error because it overfits or choose the wrong questions.
Yeah.
Have you noticed?
Yeah, so it's also a good question. So here, because we try to make this autonomous, so the AI is creating their own assessments. So I think there are some limitations to that. And ultimately, I think the real feedback comes from we do the wet lab experiments and then provide those experimental results back to the AI scientists. And I'll come to in a couple of slides of the wet lab validation we do and how we give that back to the agents. I think that's sort of the longer term feedback. I think the reason why we made this process autonomous is that this enables the agents to learn about topics beyond which I could teach the agents. Like I don't know too much about particular areas of immunology, but the agents, because they do their own learning and assessments, then they can learn, become a better expert in those domains. They're not limited to what we have expertise in. Yeah, so it's a good question. So I guess it's maybe also a bit like this, academic labs, right? So sometimes maybe the professors do not know as much about as the individual students with experts in a particular domain. But the job of the PI agent here is to really provide some structure to the group meeting, right? And if it sees like some ideas that repeatedly come up across multiple meetings, then try to come to a way to resolve those disagreements and also come to a consensus. But the PI here doesn't it's not like the PI agent try to do a lot of the scientific discovery itself. It's really mostly providing some guidance and allowing the individual scientist agents to do their own work. Okay. So now that we've seen how the agents interact and then so how they go through the training school, I'll come back to this running example of to illustrate the kinds of research and the science that they can do. Right. So recall that our project, the first project we gave to the agents to basically design binders to the COVID variants, and they decided to design nanobodies. Now, the problem with nanobodies, as they pointed out, is that there's not a lot of data to fit these nanobody models. Right, so to mitigate that, the agents decided to create a computational pipeline to purely model the structures of these nanobodies in silico. So they created this pipeline here, which is actually pretty interesting and quite sophisticated. So first, the agents decided to use this tool called ESM, which is the protein language model, to come up with some kind of candidates, right? So ESM, what it does is that it takes its input, an amino acid sequence, then computes a score to say how likely is that sequence to form a stable and functional protein. So that would be like a first filter in this pipeline, right, from the virtual lab. So then the agents would take the top candidates from the protein language model and then pass that into AlphaFold together with the spike protein of the target COVID variant. And then AlphaFold generates this three-dimensional structure, right, of how that nanobody might bind to the target spike, right? So then the agent will actually look at that three-dimensional structure and then pass that three-dimensional structure into another tool called Rosetta, which does like a more physics-style molecular dynamics simulation to relax the structure, refold it to estimate the binding affinity. So these three different tools, they all offer somewhat complementary but different information. So the agents will look at the output from these different tools and come up with a holistic score to prioritize different nanobody candidates. And then they would iterate through this process multiple rounds to come up with the top candidates. So what I found quite interesting about this pipeline, this workflow, is that this is actually different from what people have done before, right? So it's pretty different from the pipelines that people have previously published. And the agents also have done some interesting modifications to these different tools. Like, for example, it modified the scoring functions for this ESM protein language model compared to what people have previously done. So I think there's some sort of combinatorial creativity that agents could do by taking different tools that people have previously created and then combining them into sort of new pipelines and new workflows, right? And maybe modifying them a little bit, and then using that new workflow then to help it to generate, in this case, to study the proteins and to optimize the proteins. Yeah. Yeah. So it's the different individual tools have been used in the community, right? But I think the particular way that they combine these different tools together is different from what people have previously published, from anything paper that we've seen. Right. So for example, previous papers would, for example, just use ESM by itself to come up with some of the candidates for scoring mechanisms, or maybe they will combine AlphaFold with Rosetta, right? But the way that it actually uses all three tools with this holistic mechanism is different. Now, people can argue about, okay, how novel is this, right? And I think that's maybe not the main point here. It's like the agent's not trying to create its own tool from scratch, right? I think this is a different notion of combinatorial novelty or creativity, whereby the agents are able to use existing tools and then combining them into sort of a new workflow that's adapted to solve its problem. But I think sort of the ultimate measure, right, is really in how well does this actually work in solving this particular project, right? And so in this case, basically the agents developed this pipeline, they implemented it, right? And then by running this pipeline, they came up with a set of 92 nanobody candidates. And this is actually already quite useful because the space of the nanobody design is actually exponentially large, right? So the agent said, okay, you don't have to test all of these millions and millions of nanobodies, just test these 92 that it thinks are the most promising ones. Now, because they can't do their own physical experiments, right? So then that's where the human AI collaboration is quite critical, right? So the agents will tell us, here are the 92 nanobodies, and then we can, with our experimental collaborators, then we mate and synthesize these nanobodies and test them in the real wet lab. So here are two examples of nanobodies designed by the virtual lab agents. And the bottom here basically shows the wet lab experiments to test how well do they actually bind to the different COVID variants. So I'll just explain the different curves one by one, right? So the bottom red curve here was the open red circles, right? The lowest one here. That's where if we take the existing nanobodies, they're actually designed by human researchers, and we test those against the recent JN1 variant, which started to spread last year. So you can see that the existing human designed nanobodies that's not actually bind to the JN1 variant. And then the second red curve was the solid red circle is the one that's designed by the agent, right? So that's the new nanobody. And you can see that that actually does show more promising binding to this new variant. And then the two blue curves are basically where we test the two nanobodies against a very different strain of COVID, right? Basically the one from 5 years ago from the Wuhan strain. You can see that the solid one, the one designed by the agent, also shows more enhanced binding to this very different strain of COVID. And I think that's kind of promising, right? Because here there's the same nanobodies designed by the virtual lab actually starts to show binding across this whole broad spectrum of COVID. So even as the virus evolves, it accumulates hundreds of mutations, the same binder actually still shows consistent binding, which is was a pretty challenging problem. What we're not showing here, but we also did additional experiments to test these nanobodies against other proteins, like your host proteins against other viruses, right? And they don't bind to these other viruses or to the host, right? So they're actually specific for the COVID variants. And it's also quite useful because you want to have that kind of specificity. And what's kind of interesting is that then we can also give these experimental results back to the virtual lab agents, right? For example, we give these figures back to the agents, and then they could interpret those experimental results, and they actually further refine and optimize their computational pipeline, right? So there's actually a feedback process whereby the agents can continue to improve as they get more experimental data back.
So it sounds like the AI dynamic here. It's like the AI discuss and come up with like use experiments, and the human in the real world the experiment and kind of report it back to the AI itself.
For this experimental part, yes.
So I guess I do think like it's really impressive that it looks like the experiment is working, but like is there a chance like maybe the AI just give bad experiment ideas? And I guess how do we safeguard against that, since the AI is good at giving like hundreds of experiment plans, right? But the human, obviously, we're trying to save time. So is it just like the human judgment to decide whether we do actually carry out the wet lab, or like we have any safeguard that the AI is going to give us good experiment?
Yeah.
To actually,
It's a good question. I mean, here, one thing we do is that we told the virtual lab agents upfront, right? Here's how much budget we have to do the wet lab experiments, right? So that constrained them so they can't come up with hundreds or thousands of designs. They have to say, okay, here's 92 designs because that's how much budget we have, right? So they have to be quite selective themselves, right? But also the agents are able to provide rationales and explanations, right? Because here, they communicate to each other through natural language, right? And they explain their reasonings, right, in natural language. So that also makes it easier for the human teams to look over their reason and say, do we agree with their rationales and their justifications? But ultimately, the human scientists can decide to go back to the agents and say, try something else, right? So the decision still belongs to the human teams. Yeah. Yeah, so we synthesized each of the 92 nanobodies, and we tested all of them. Right, so there were, I think, about 4 or 5 that showed some promising binding, and these are the 2 that showed sort of the best binding across the different diverse strains of COVID. Right, and so I'd say that's actually pretty good success rate, right? Because in the conventional kind of designs, people often will test for a lot more candidates, right? Usually hundreds or thousands of these nanobodies, right? And for example, the best comparison is the human design nanobodies here, which have gone through a much more extensive process design, and they don't actually show as good bindings as the ones designed by the virtual lab. Great. So just to summarize this part, right? So we actually made all of the code for the virtual lab open source. Right? So it's a part of this paper. So people can use it actually to tackle different kinds of projects. Right? So it's not limited to designing proteins. Right? You can use it to tackle other kind of scientific problems in biology, medicine, but also in other domains like physics or chemistry. And one learning of this is that we found this that actually when you have multiple agents with different background to work together, that often led to more robust and also more creative results than if we try to have a single agent to solve the project itself. Right, and this happens because, for the reason we mentioned before, the agents can also have these debates and disagreements that led to more robust reasoning process. We found it to be quite helpful to basically build various toolings for the agents. For example, the agents each have their own memory mechanisms. They can remember what they previously discussed in their previous group meetings and one-on-one meetings. So that's quite useful. And we also built like a sandbox environment where the agents have their own virtual environment where they can run tools like AlphaFold locally. So that makes it easier and faster for the agents to modify those tools and also to to debug and then to self-improve. So the last part I think is quite interesting is that I think the agents also start to develop their own social dynamics. Right? So when human teams work together, then some, maybe some people talk more than others and they have louder voices, and that affects changes the dynamics of research. So similarly in the agent teams, right? So some agents speaks more than other agents. That also changes the outcome, right? The kinds of research that they do. But one thing that's very much easier to do with the agents than with humans is that we can change their dynamics. Right? For example, we can change the order with which different agents speak. We can also change their personalities, right? I can make some agents more talkative than others. And we can do all of those experiments in parallel, right? So almost as like a petri dish of social science to see what combinations of team sizes and personalities actually leads to the most robust and creative research. Right? So you can't really do that easily with human teams. But one of the nice things with virtual labs is that we can do all of those benchmarks and evaluations in parallel. And maybe that can also teach us something about how to do better science within human teams. Yeah. Yeah. I think a lot of that also depends on what is the goal that we give to the agents, right? Because in this case, I would say the creativity of the pipeline is not maybe the ultimate metric, but it's really the creativity and the efficacy of the nanobodies, right? So these nanobodies are creative and novel in the sense that they're actually quite different from any existing nanobodies, right? So they're at least like an added distance of 3 or 4 away from any existing nanobodies. So in that sense, these are actually quite novel designs. And the fact that they actually work is probably the top priority for the agents. Right. So if we had asked to give the agent a very different objective, like come up with an entirely new pipeline, right? Then the goal is different, and I think they would have came up with something that's very different. Right? But that's not the goal here. It's true. I guess a lot of the existing nanobodies are still the way that people design them would be, with usually within like a 1 or 2 edit distance, right? They will take the previous nanobodies that worked well, right, and then try to find maybe 1 or 2 mutations from that can bind to this new variant, right? So here the agent's actually explicitly searching for a much larger design space. Yeah.
How automated was the simulation pipeline?
Oh, so this pipeline, so this is fully automated, right? So it's it's implemented by the agents, and they can just run this pipeline.
The code agent actually ran.
So there's the debugging process. So that a lot of the debugging happens when the coding agent would meet with the PI agent to review the codes. Yeah.
That from 92 potential ones that the virtual lab recommended, and you validated them, and those were the 2 best ones. Do you, by any chance, recall where they rank among the AI suggestion? Was it like among the top 5 recommended, or where is it? And I also wonder if you ever run like a negative control lab and say, here are 5 agents, we're just high school students. You have your discussion, you come up with another 92 suggestions. I will tell you I want nanobodies, but you're just high school students and you read something. You recommend 92 and we validate your 92 as well and see whether they do any better compared to virtual lab of supposedly expert.
Yeah. So both are good. So I think these 2 here, I don't remember the exact ordering, but they're near the top, like I think within the top 10 or 12 of the 92 by the agent's own scoring mechanisms. And we did do other oblations, right? So one oblation would be we basically just have generic agents, right? So we didn't tell them they're high school students, but they're not experts, right? And we have them basically come up with different, try to solve the same problem. And those agents actually run into a lot more problems. For example, they will even have a lot more issues like in writing the code or in finding the right tools to use. Okay, so good questions. So in the second part, I want to build off this and explore a bit of how the agents can do more data science, right? So here we build these data scientist agents that can take as input these quite complex data sets, include genomic data sets, and generate Jupyter notebooks to analyze those data sets and then create research reports. And the motivation for this is that there are a lot of parts of biology now that's generating quite complex data, for example, like the single-cell RNA sequencing data, right? And those are quite challenging for end users to analyze because the data is quite complex, but also the space of tools is also quite large, right? There's over 1,000 tools for analyzing single-cell data, and a lot of the biology users, they don't quite know which tools are the best ones to use. So as a result of that, people might only apply some of the standard analysis to this rich data sets, which might only scratch the surface of what you could learn from those data sets, right? And we wanted to see, can we basically have like an AI agent to help us to explore new analysis, maybe a little bit less conventional analysis that might have been missed by the original researchers. And so here we created CellVoyager agents, kind of like a data scientist agent, right? So this agent would take as input the data sets and any write-up of your previous analysis. And then would generate like a new analysis with new figures and reproducible code to hopefully provide some new insights that were otherwise have been missed. So briefly, the way that this works is that, so first the agent will actually prepares and explores the data set to understand what is in this input data, then does a deep research to understand what is the broader context and how did people have previously analyzed similar data sets before. And then it prepares the environment. In this case, it would be like a Jupyter notebook with the relevant tools and packages. And based on that, then it starts to generate a set of hypotheses of interesting new analysis that you could do actually on this data set that was not done in the previous literature, right? For each of these new hypotheses, it would generate like an analysis plan and then execute that in a Jupyter notebook by writing code, generating figures, and then interpreting those figures to decide on what to do next, right? So it would be like a sequential set of analysis to go deeper and deeper into the data sets. So here's the example Jupyter notebooks as created by the agent, right? So each of these notebooks would have a series of these organized code blocks, right? So it would always start with like a description of what is the hypothesis or analysis it's trying to do. And then we write a piece of the code to implement that test, which by running that code will generate this figure, and then we interpret the figure to figure out what is the learning and then decide what to do next. So the way that we try to validate this is to basically we give the agents previously published papers and the corresponding data sets. Then we ask the agent to reanalyze that data set, but come up with new insights that were missed by the original authors. And the way we validate that is to basically give the report from the agent back to the original authors and have them double check that first the analysis was done correctly, and then second, that these were indeed new insights that they had themselves had missed in their original analysis. So we did several of these case studies. So here's one of these, which is basically having the agents to analyze brain aging data sets. So here it's basically single-cell RNA sequencing data sets across the brain from different ages. So the original study focused on basically building machine learning biological age clocks to predict the age from the transcriptomics. And here the CellVoyager agent generated 5 new analyses. And then when we give those back to the original authors, they found that all the analyses were done correctly, and 4 of the 5 were quite interesting and different from what they had thought of before. So here's one example of these kind of analysis. So these are the figures that are generated by the agents. So what typically the agent will actually do some data exploration and visualization first, right? So here's visualizing the single-cell RNA sequencing data. And then it quickly dove into this hypothesis about looking at transcriptional noise and age. Basically found that cells that are biologically older tends to have higher transcriptional noise in the sense of having higher transcriptional entropy. So this is a pretty interesting correlation then between the entropy and age across these different cell types. So this has been shown in other organs, other tissues, but people haven't observed this before in this part of the brain called the subventricular zone of the brain. All right, so the agent actually, made this discovery that was sort of missed by the original authors. So it's not super surprising, right? So it's concordant with what people have seen in other organs, but it is kind of a new finding that also contributes to the aging literature by expanding this observation to these new tissues. Okay, and then subsequently, then the agent also went out and found additional data sets, right, from other studies and download those data to further validate this finding that there's this correlation between transcriptional entropy and age across other animals. Yeah. Okay. So the last part I will wrap up into mention is this quite recent idea that we're exploring that we're calling paper to agent. It's sort of like a way to think about reimagining what is a unit of scientific communication, right? So the standard way that people communicate research is by writing these papers, which are essentially passive artifacts of research, right? And usually, the reader, the onus is on the reader to understand what is in the paper. And if the paper releases some code, right, or data, then it's also often pretty challenging for the reader to figure out how to adapt that code or to use that data. So we thought, okay, so instead of having these passive papers, why don't we turn these into active agents, right? So that you now have an agent, you can ask the agent questions, right? You can ask the agent, for example, use the tools developed in this paper and apply that to my new data, right? The agent will actually run all the analysis itself and then generate and produce those results, right? So in that sense, then the paper agent is sort of like an expert of that paper that learns how to use that paper and use the code in that paper and then help the readers to apply that to their own problems. So the way that we build this paper to agent is that first, the main challenge is that if you just ask the language model directly to use the paper or to read its code base, it's often not very reliable, right? So to address that, we created this automated workflow to basically convert each paper into a reliable MCP or a model context protocol, so that then the downstream agent can actually use that MCP as a resource. So the way that this process works is that first, we have a multiple sub-agents, right? So first they will identify the code in that paper, right? Read the GitHub page, and then extract all the tools that are relevant from that code base, but also build the environment that are relevant for running that tool. And then the sub-agents will actually create a set of unit tests to test how well can we actually implement those tools in the MCP, right? If it does not do very well, it'll actually self-improve and self-refine. And the output of this will be like a robust MCP server that basically captures all the insights that will come from the original paper. Right, so this MCP server would have tools, would have resources, which could be the different data sets in the paper. It could also have different workflows that captures how the authors in the paper had used these tools before. And then we can expose the MCP server into like a downstream chat bot, could be Anthropic or OpenAI models. And those chatbots can then use the MCP to become like an expert in that particular paper. So that's how we go through this paper to agent process. So here's an example, right? So a lot of people here would use these tools like ScanPy to help them to do like single cell analysis. It's quite widely used, right? So then with paper to agent, we can basically create like an agent version of ScanPy, right? So the agent here basically knows all the different tools in ScanPy. And then you can just ask the agent in natural language, like, here's my data, do this analysis for me, right? And the agent will actually use all the different ScanPy tools and then run that analysis and generate the results. And in this case, right, it's able to basically exactly reproduce figures analysis that human experts will be able to do by manually running these tools. So here's another case study. There's a recent nice work from DeepMind called Alpha Genome, right? It's also very useful for interpreting, doing functional interpretations of variants, right? It's a pretty complex set of tools. So here, by running paper to agent, then we can basically create an Alpha Genome agent, right? then you can just interact with the agent through natural language and say, okay, use the alpha genome to interpret this particular variant and try to identify what are causal genes for LDL cholesterol. Right. Then in this case, the agent, because it has all the it knows about how to use alpha genome, right? So then it will actually create the workflow and then run the relevant tools in alpha genome to generate these intermediate figures and results, and we interpret those figures, and in the end come up with some findings like, oh, in this case, it thinks that the causal gene would be sort 1, right? So this makes it much easier for the end downstream user to basically directly use the insights and tools from that paper without having to download anything themselves or having to set up the relevant environments. So our goal here for this is to basically use these like paper to agent to convert, basically passive artifacts of research like papers, right, into these active agents. And that, I think, would make it much easier for the downstream users to be able to leverage the knowledge and methods and data from these papers, right? So now they can just essentially have an expert that can help them to apply these methods from the paper to their own problems. And I think this would be a kind of a nice way to disseminate knowledge, right? So instead of having the onus often be entirely on the reader to understand what's going on, right? So essentially now we have an assistant, like a AI corresponding author of the paper that's always available to help the reader apply the method to their new problems. And also these paper agents can also collaborate, right? So you can imagine now we're creating more and more of these paper agents. You can imagine like the agent of a new methods paper can then interact with the agent of a new data resource paper, right? Then those two agents can also collaborate and apply that new method to that new data to generate new insights, right? So I think there could be a lot of opportunities for how these paper agents can facilitate research. So I'll just wrap up by also sharing like we are putting a lot of these different pieces together by organizing a new experimental conference called Agents for Science. So this is the first conference where both the authors and also reviewers are AI agents, right? So you have to be an AI to submit to the conference. Humans can be co-authors, but AI should do most of the work. And then all of the submissions also be reviewed by other AI agents, right? And then we'll have humans to do a final assessment of the top papers. Right. So this is an experiment. So all the papers, submissions, and reviews will be publicly available as a way for the community to learn about what are the strengths and weaknesses of AI in doing research and also in reviewing papers. Right. So the deadline is coming up next week. So you still have time. You can get your agents to work and then try to do some interesting research and submit to the conference. Yeah. So I'll stop here. And want to especially acknowledge my students that led these different projects. So Kyle Swanson for the virtual lab work, Sam Albert for CellVoyager, and Jiachen Miao for paper to agents. And I hope through these examples to illustrate how why we're quite excited about these AI as sort of co-scientists to help us across this discovery process. Yeah, so I'll stop here and happy to take any questions.
Each LLM to put a and answer reliable questions reliably. And we found that it's sort of not critically sort of unreliable answers, they hallucinate something. And of course, there's a broader discussion of this issue. It seems like you've been able to get around a lot of those issues. Right. So could you say how you're able to avoid all the hallucination problems, the errors that tend to crop up at the bottom? What is it that we should be thinking about in design?
So it's a good question. I think the current agents, they're really good at there are two broad classes of things. One is using tools. The second is in writing code. Right. So if there's a way to frame our research problem through some combinations of coding and tool usage, right, I think that's a great way to reduce hallucinations compared to if I just have the agent try to think through in English what's going on in this problem. So so in the protein design case or in computational biology, I think that's pretty amenable because there are a lot of existing tools like AlphaFold, right? So when the agent design a protein by using AlphaFold, then that's much more reliable when the agent then compared to if the agent tried to just hallucinate the protein itself, right? So I don't know about the legal literature. So maybe in there are less available tools for the agents to use, right? Maybe that's one of the differences. Great. Yeah, I think that's quite interesting. I do think there's also value in having the negative results publicly available, which is why, for example, for our conference, we'll make all the submissions, both accepted and also rejected submissions, publicly available. So that I think it's actually just as instructive to figure out what are the failure cases of the agents, right? Like what are the problems where they cannot solve, right? Where they have a negative results. So I think that will actually be quite interesting. And if some of those their discoveries from the agents also match the human failure cases, that's also quite useful.
Part about running standby using an agent. So one of the issues we run into is that there's already a level of abstraction when you're talking about standby. That students run the code, we don't know what hyperparameters were default happens. And then with the AI agent, there's another level of abstraction. So now when the result is not what you expect, how do you go and kind of debug so you don't get it?
Yeah, so that's a good question. I think there's always this kind of trade-off between how user-friendly, let's say if a biologist doesn't know coding, right? Then how much, well, what's the right level of abstraction for that particular user? I guess the way that we've been thinking about this is that under the hood, the agent will always generate things in the Jupyter notebook, so that if somebody does want to go back in and then figure out exactly what's going on, they can just rerun that Jupyter notebook and check exactly how to reproduce each of the analysis and figures. So there's that level of very in-depth transparency. But if the end user doesn't want to look at the code or doesn't know how to look at the code, right, they just want to say run a standard analysis and tell me the end results, then the agent would just produce the end result back, right? But it would still produce that trace that people can go back to and audit. Yeah, so it's a good question. So I think there is some complexities in that if you try to ask the same agent to learn from multiple different domains, then there is some forgetting and it could actually some interference, right? And people have to be pretty careful with setting like the right data recipes, right? To make sure that the agent does not forget what they learned before when it's this additional learning. Which is also why in our case, we find it easier to train the agents to have more specialized expertise rather than having like a single agent to learn all of these topics by itself. Then we have an immunologist agent, or I have like an AlphaFold expert agent, right? So those are more manageable chunks of knowledge for the agents to ingest.
Every now and then they're right. So when they're right, every now and then it's a very useful idea. I feel like with some of the agent descriptions, like they start a bunch of things that every now and then they're right. But there's other things that aren't, like the 90 parts, some of them. So I guess the question is, able to find a way to sort of express some level of uncertainty of non-finance, things the agents are producing, so that if you're able to get a sense of, okay, I'm saying this, but what I'm saying is I'm pretty sure. So this issue of uncertainty.
Yeah, so that's also, I think, a really interesting and generally important problem. There are two levels of uncertainty here, right? One is that because the agents actually use different tools, right? Like when it uses AlphaFold, AlphaFold itself also produces uncertainty, right? And then there's an additional question like how calibrated is AlphaFold's uncertainty, but at least there are ways we can do it to calibrate that, right? So I think this is where tool usage does help us in that, because the tools also provide some intrinsic eccentricity themselves that actually provides additional layer of observability that the human can look at and say, okay, yeah, this protein scored very high by the agent, and also AlphaFold is very confident. So that gives me more confidence that I should test that design. I find that level, like tool-based certainty, to be much more reliable than if I try to quantify the log odds of the agent itself in producing a particular sentence. How do we evaluate the different ablations? Yeah. So we have to identify some sort of let's say proxy metrics, right? Like one proxy metric could be like how many bugs in the code does it write, right? When it's trying to implement this computational workflow, like how quickly can it implement it without having bugs, right? Another way we evaluate this is to basically have blinded human evals. Essentially, we have different versions of the virtual lab teams generate research plans, and then have human experts to evaluate which plan do they like better, right? Of course, the human experts also have their own limitations and biases, but that also provides like some sort of proxy. Yeah, that's also a good question. Here, for example, when we look at the generated nanobodies or proteins, so there is a more objective metric of novelty. We can just see how different is this protein that's designed by the virtual lab compared to any existing protein that people have seen before, right? And there we know that like the nearest neighbor is like 4 edit distance away. So that's a reasonable notion of novelty for the end design.
The difference is that, like adding too many documents.
Yeah. Yes, great point. Yeah, so we did some experiments. We just tried to have larger teams, and we found that when you have too many AI scientists in the team, that also degraded the performance. So sort of like too many voices. So in these experiments, we found sort of between 3 to 6 agents. It's sort of like a sweet spot. Yeah.
Agents. At the beginning, if you have 5 parallel virtual labs and each one of them has computational biologists, we can think of those 5 computational biologists as more or less exchanging, right? But then there are 5 labs, they do different things, they're different agents. Do they keep their memory, know what they did, and get the feedback of how their work is taken into action and what the results are, so that later on there are different computational biologists, each of their own history.
Yes, yeah. So each agent has its own memory mechanism. So they remember what did they talk about in their one-on-one meetings and also in their group meetings. We also basically have the PI agent to periodically summarize the current status of the project for all the other agents, right? So basically the PI agent will say, here's everything that we have discussed and done so far, and here's the next set of steps, right? So it's a way to basically level set and to sync up across all the different agents.
What kind of data are unattainable or unreachable by these advanced, say, image? Often there are controversies in the literature or retractions. Images are not the supportive particular hypothesis. Are there certain types of data? And let's say single cell RNA-seq is very digital and therefore very amenable to this analysis. But are there types of data in the biomedical literature that are sort of unattainable or
Yeah.
reachable or accessible by the AI agent?
Yeah, so good question. So for example, when we turn a paper into an agent, with paper to agent, right? So that agent then relies on the data that has to be publicly available or released as a part of that paper. So let's say if you have like data, a paper that uses a lot of PHI data, and that data is just not accessible, right? Then the agent will not be able to provide help on those hidden data sets, right? So the agents, for example, is I think the paper agents tends to be more useful currently for the biological papers where the data is actually released together with the code, because then that paper agent then becomes has the full context and the full knowledge, then it can become like an expert user, an expert applier of that data and methods. But for the more medical ones, if the data itself is not released or the method is not open source, then there's nothing the agent could can really do. Okay, great. Well, thank you very much.